Our next speaker is Dr. Iain McGilveray. Dr. McGilveray received his BSc in pharmacy and a PhD in chemistry and pharmacology of antibiotics in Glasgow, Scotland. He received postgraduate training in antibiotic chemistry at the University of Illinois and in biopharmaceutics at the University of California. He is a fellow of the American Association of Pharmaceutical Scientists, serving as chairman of the regulatory section in 1999 in the Canadian Society for Pharmaceutical Sciences. He was a member of the Board of Pharmaceutical Sciences for the International Federation of Pharmacy and the founding member of the first chair of the Bio International Conferences. During over 30 years as a research scientist at the Bureau of Drug Research, Health Protection Branch at Health Canada, Dr. McGilveray contributed to over 150 research publications in analytical, methods, biopharmaceutics, and pharmacokinetics. Prior to leaving the Health Protection Branch, he was chief of the Biotherapeutics Division of the Bureau of Biologics and Radiopharmaceuticals, involved in the review of recombinant biotechnology products, including hormones, recombinants, and monoclonal antibodies. He was one of 3 acting directors of the Bureau of Drug Research in 1996 and 97. He has been a regulatory biopharmaceutics, pharmacokinetics consultant to the pharmaceutical industry since leaving Health Canada in 1998. Dr. McGilveray has been advisor to the WHO and the US FDA. He was awarded the Distinguished Career of the Drug Information Association in 1998. From 2001 to 2006, he was adjunct professor of medicine at the University of Ottawa. So I think this is very timely following Dr. Law's presentation to hear Dr. McGilveray, and please join me in welcoming him.
Thank you very much. Is there a pointer? No? Guess I'll just have to use my finger, right? But it might be helpful later. Anyway, I can start by thanking Pregmed, Betty Ann Fuse in particular, and yourself for inviting me to this. And I think it's a very interesting area because you're right. We are not doing everything the right way, but that takes time for government to move. So, my disclaimer is that I'm not of the government anymore. I'm an independent consultant to pharma, and that includes Duchenne, who gave me some data. Manon is in the audience today, who helped me with that. The original title of this, I believe you've changed the title, was Clinically Relevant Pharmacokinetic Changes in Pregnancy, which was talked about. And that's my second disclaimer. As has been said, there are very few bioequivalence studies in pregnant women. I don't know of any that are actually published for obvious reasons. But I will talk about pharmacokinetics briefly. I'm going to talk about alcohol, the oldest drug, because it has, very briefly I'll talk about it, it has some information for us about history of how things happen with knowledge. Bioavailability and bioequivalence are subtopics from pharmacokinetics, and there are regulatory guidances, mainly from FDA, but you talked about some European guidances, and I haven't dealt with those. There's not much, as you've said, in Canada for this area of women and pregnancy, but you say there's a new one that they're looking at. Individual bioequivalence guidances are all from the US FDA. They have a great many. I haven't many examples, and I'll end up with some concerns and inclusions to say that the situation may be getting worse. The semantics, I haven't actually talked to Dr. Sullivan, but she has took the wind out of my sails when I started this, and I read her paper to say that I shouldn't use the word gender because it's cultural. And I don't like the word sex too much because it's got that nudge, nudge, wink, wink factor attached to it. So, but I do notice that most dictionaries do note gender and sex as synonyms, and I will be doing that, as have other authors before. But maybe we need a new word, so if you can find it, please do. So I'm sure on Friday afternoon you want to see differential and calculus equations, but I'm not going to do that. But pharmacokinetics have complex models, as have gas prices, as you know. I'm going to start with this kind of complex one, but to try to simplify it. When we're talking of bioequivalence, we're really talking about this end, what happens from the drug dose to absorption, and are there differences in absorption? The unfortunate thing is, for what we measure, which is mainly plasma levels, sometimes whole blood levels, and occasionally urine, but we're looking at this great mess of stuff that's affected by metabolism, by binding, which does change in pregnancy, and is certainly different in some extent with sex hormone binding globulins and things in men and women. And there are some differences, as we've seen, in metabolism and secretion. But this is a big mess that we have to sort through to find out what happens at this end. And it is difficult, and there's a lot of variation, more with some drugs than others, and more with some studies than others. So I get to the regulatory guidelines that I looked at. There is a guideline that came out in '93 that did start more women being involved in clinical trials. But before that, there was certainly deficiency. So for drugs before then, they've not been well studied in women, and that's the problem we have now. There's also a guideline, Pharmacokinetics in Pregnancy. I think it's a draft guideline, and they have recommendations there, and we heard Europe has that now. But the only thing that I've seen from Canada was that thing, and some of the people who worked in my division were on this committee that came out with it. The policy issue was inclusion of women in clinical trials during drug development. These more or less say we need to have more women in the clinical trials. So the reports, looking at this, Yang and the colleagues from FDA published a second report from FDA, actually, about participation of women in clinical trials for new drugs over the years 2002. And their sort of big conclusion is overall participation by women and men was comparable, improved since the prior report, which dealt with the years 1995 to 99. Still, there was an underrepresentation of women in early phase trials, and for example, cardiovascular products. That continues to be an issue of concern. For new drugs, the studies in women and men are reported in the ICH Common Technical Documents, and particularly you'll find these in the clinical pharmacology summaries, Module 2, but of course Module 5 has the details of the clinical studies. And there are often population pharmacokinetic studies in patients that are done in phase 3, and there's always some analysis by gender there. The ICH E5, which is about ethnic differences, also includes gender as an intrinsic factor that should be looked at, but that's when you come to special populations. So I had the first report from the FDA on women and pharmacokinetics, and Schwartz in 2003 did have a review suggesting that absorption is not significantly affected by sex, but rates may differ, maybe slightly slower in women, rates of absorption. And as someone had noted, in pregnancy that might be worse. And for CYP3A substrates in particular, maybe somewhat higher, so there'd be higher exposure for women compared to men, higher AUC, lower clearance. The role of sex in the pharmacokinetics of genetics is much more complex, though, because we don't know where it fits with age, disease, social habits in the clinical setting. So needs more study. This is a common phrase said, give me more money. So anyway, this is an earlier study that is an interesting conclusion, gender differences in pharmacokinetics. For the majority of investigated drugs in recent years, no or any very minor gender differences could be detected in pharmacokinetics and pharmacodynamics, and I find that strange because I know that in psychoactive drugs, women seem more sensitive, starting with alcohol and working to antidepressants. I think that they are more sensitive despite the kinetics. And their clinical significance maybe seem very limited, really linked to success or failure. And that's because many drugs have a wide, they're not narrow therapeutic index drugs, many drugs. Hence, it is necessary to include, it's necessary to include women, but questionable whether they should be in early stage studies. So that was their conclusion. Yes, let's have women in, but not too early. Maybe that's the correct one, I don't know. Dr. Selden in here has a very fine review and notes that males and females may differ in specific drug pharmacokinetics and dynamics, and therefore it's essential to understand those sex differences in drug disposition and response. So obviously this has to be studied. So all including regulators agree that such studies are important, but at what stage? Except for drugs used only in women, such as for breast cancer, caution prevails, and it's usually not till phase 3 studies. And studies in pregnant women are obviously rarer, but there are some in antivirals and antimalarials, as the concern is that fetal transfer might help protect the fetus. And I looked up, most of the studies are on these types of drugs. So I said I'd talk about alcohol. It's been known since antiquity that women are more susceptible than men to alcohol effects. And why? And of course, at this a society meeting on Wednesday, Dr. Bryan gave an excellent talk on fetal alcohol syndrome and his animal models and things that are involved there. And we're beginning to understand more about that. But we still don't know if it's pulse, doses would be bad, at what time in the, whether it's first, second trimester or first 2 weeks, that alcohol would have this effect. This, I did probe Dr. Bryan about it, and we don't know the dosage or the actual time of dosage when it would be effect. So it's still, we don't know a lot there. But some effects are due to the body mass, with the higher blood levels being common in women, a larger exposure. And a small 1996 study did indicate there was a significant 28% higher in men, but it's more complex than that. And a 2001 report said the gender difference in alcohol levels is due mainly to a smaller gastric metabolism in females because of significantly lesser activity of the main metabolizing enzyme ADH, rather than differences in gastric emptying or hepatic oxidation of ethanol. And in the last, more recent review, I guess, says that, I haven't actually given the reference, sorry about that, I'll get it though. There are many other things influencing this effect in women than just body mass and these other things above, but particularly an important determinant allelic variants of the genes encoding the alcohol metabolism enzymes ADH and ALDH. Thus, it remains that some women are less susceptible, and even now, why is not fully understood. And this is a drug that's been there since antiquity. We don't know everything about it yet. So more recent drugs, we're in worse shape there. Let me get back to the bioavailability I'm coming to. This is a simplified ADME, and we are, of course, as I said, interested at this point on the absorption. The liberation is in vitro, in vivo, and then it goes through the body, as I'd said. Mainly metabolism is what is dragging out the drug. And so we're interested on the formulation, on the area under the curve that comes from that, and that's the main exposure metric. The only equation I have up here it's a simplified one, but it's total body clearance equals dose over AUC. So there's a relationship between clearance and AUC, and EUC is, they're both exposure metrics. And as I say, metabolism is a major influence, and it's studied by looking, first of all, where you can, at the absolute bioequivalence. I won't spend time on this slide, but it does show a schematic of what happens to a solid oral dosage form at release prior to absorption, absorption being the key determinant of bioequivalence, really. And here is from Goodman and Gilman again, a simplified look at the bio side where absorption occurs. And the early things where it was mainly by diffusion, affected by the pH and the pKa of the molecule, but it's more complex in that they're transporter mediated, can be affected. But this is still the main determinant of absorption, getting it soluble and then the pKa. So I'm going to quote from the guideline I was associated with, and they're still trying to revise this. I hope the revised guideline does come out soon. But this is Part A, it's of the Bioavailability and Bioequivalence Guidelines, that John Rudy was a chair, and my group contributed much to the science behind it. The bioavailability is a PK attribute. It's defined as the rate and extent of absorption of a drug into systemic circulation. However, it has to be assessed by serial measurements of the drug in the systemic circulation and provides a plasma concentration curve, and from which important PK factors can be determined. Now, the simple ones, and they're not model dependent, are area under the curve and the maximum observed concentration. They often look at peak time, that's the Tmax. They talk about when Cmax is reached, but that is not used in the regulatory sense very much. The area under curve provides the estimate of the amount of drug absorbed in the systemic circulation, and the Tmax reflects the absorption, and to some extent so does Cmax. So EUC is regarded as a measure of total exposure, and Cmax the rate of exposure. So comparison of the EUC values comparing intravenous with oral, that is oral over IV, gives you the absolute bioequivalence for most drugs, and therefore you can get something like 20% only by the oral route, because the rest is lost to metabolism and other things, first pass being the thing that causes variability. And the comparison of test and reference product becomes the bioequivalence situation. An estimate of comparative bioavailability, test and reference are said to be bioequivalent when the profiles are similar according to statistical assessment and by meeting stated standards. In Canada, the general standard for EUC and the US is 90% confidence interval of the geometric mean ratio, and that has to be within 80 and 125%. In the US, they also have that standard for Cmax. In Canada, we have a rather wider standard. It's the geometric mean ratio for most drugs. But in Canada, they have isolated out the critical dose drugs. Thyroxine is now one of them. It's not in the list, but it is. And warfarin, obviously, and phenytoin. But these drugs, you're required to have a 90% confidence, 90 to 113%, and then there is a Cmax. It's the same, it's 80 to 125. So there's a tighter standard for some drugs. The Americans have just a general standard for everything. So the important thing, and we have talked about it earlier today, the projection that came out when bioequivalence was started about 30 or more years ago, it implies that the drug product can be expected to have the same systemic effects, both therapeutic and adverse, as the reference product when administered to patients under the conditions specified in the label. For all these years, the premise was and is that healthy volunteer studies can support this assumption. And these are crossover studies, I would say, and that's the important thing to note. Drugs with uncomplicated characters can usually be tested in normal, healthy volunteers. The investigators should ensure that female volunteers are not pregnant or likely to become pregnant during the study. That is the only real statement in Health Canada that indicates, and whether the new guideline has more about it. Certainly the FDA is much stronger in that. We recommended that if the drug product is intended for use in both sexes, the sponsor attempt to include similar proportions of males and females in the study. They want a representative group. If it's used in the elderly, they would like to see more elderly in the group. But there are differences even within these statements. But they both indicate that, yes, women, non-pregnant women should be included. So I just go to some metrics. I won't spend much. We've been over the metrics. That's the AUC and the Cmax are the main ones. But this is an important one because it's talking about the variability that we heard about today. In general, if it's a highly variable drug, is defined as that GMR-CV being 30% or greater. And if the CV is less than 50%, you can use 15 to 20 subjects. And if it's 30%, you'll maybe need 80 to 100 to meet the standard being the 8125. This is a common situation when it's not terribly variable and it's the test to reference GMR is 100%, then it's very easy to pass. This one, a very low variability, so it might be 90% GMR and it still passes. This one might have passed if they had more subjects because it's a highly variable drug. It's got 100%. This one, it could have passed if you had more subjects, but maybe shouldn't have. But these are clear failures. So you can see that the variability from the drug drives the size of the study, and we had a comment on that. You might get away with 12 because it's a not very variable drug. So this is some BE results. I won't go into that in detail, but this looks as if it might be similar, but it's too variable, and in fact, they fail the standard for EUC. They're above the level. So it's surprising that if you drew that without the bar lines, you would maybe think they were equivalent. And this was a study, the FDA of carbamazepine, a drug that you might want to worry about. This is the innovator. So all these other ones were not equivalent, but it was clearly demonstrated that they were not equivalent using the type of study that we talked about. And this was how a formulation can affect the situation. This was first discovered in 1950, something with griseofulvin. You micronize very poorly soluble drugs, you can increase its bioavailability by 50%. This has been done with glibenclamide, and they've micronized it, and they make it so that the 3.5 is actually a larger exposure than the 5 milligrams. That's an improvement in formulation, and that should be supported by bioequivalence studies if you're going to change the dose for sure. Anyway, the background, and I'm coming to the paper that was quoted earlier, and I have problems with what was quoted, and I'll try to talk about it even with what's not on the slide. Anyway, Mei Chen and the FDA did this, and I hope the FDA could do this again, but there might be problems in doing that, which I'll mention at the end. Anyway, the sample sizes for the studies were not chosen to examine the sex-related effects considered. There were 26 bioequivalence studies between 1977 and 95 where they had enough men and women to look at this. And there were less than 20 per study, the number of volunteers. The EUC in women, 71% of the time, and the Cmax, 87% of the time, was statistically higher for reference in 28% of the data sets. The frequency, well, it went went to a lesser amount if you took body mass into consideration. Women tended to have higher variability, but the results of this study support the recommendation of the 1993 FDA gender guideline that women not be excluded from bioequivalence. It seemed that the crossover study was considered efficient to detect bioinequivalence. Now, what I looked at that paper again today, there were three sort of indications of gender by formulation interaction. Two of them were transdermal patches of nitroglycerin, and I know that's a very difficult drug to analyze. I would wonder about that. But they were patches, and there might have been differences in shape and the actual rate of exposure. So maybe there was one. The other one was peculiar. It was erythromycin that had an 18% difference between men and women. Now, these were the only two drugs of that group where, when you took the other factors out, that had a difference. So it's not much to hang to say we need to look at our model again, but it would be nice if the FDA looked at more recent studies and tried this again. That would be very good. So the other thing that the FDA has done, and Health Canada probably can't afford to do, it has individual drug bioequivalence guidances. And these are quite interesting. They have 805 draft guidances. Now, that's not 805 drugs. There's maybe there's 500 drugs I didn't count, because a lot of them are kind of repetitious. There are only 153 final guidances, and most of them recommend subjects be healthy males and non-pregnant females of the general population. There are certainly some inconsistencies when you look through that, and the instructions for pregnancy and lactation certainly need tests, need standardization. They could really standardize that, I think, in there when they mention about it. About 15% of them don't mention pregnancy checks, although the women are said to be included. And I was surprised that phenytoin was among those, because it's certainly associated with birth defects. Breast cancer drugs, for example, anastrozole, vaginal preparations, and oral contraceptives, have women-only subjects and some hormones. Exemestane is in postmenopausal women. Drugs for prostate cancer and erectile dysfunction require men only. Progesterone, funnily enough, healthy males and postmenopausal females are suggested, perhaps because of the endogenous interference in premenopausal women. Tamoxifen has both men and women, as it is used in both sexes for treatment. In general, I thought the subject inclusion recommendations in the FDA are reasonable. I take up your point that Health Canada hasn't really looked at this, and it's important to do so. It would be useful, since FDA has more data, if they looked at this in more detail. So, new and old drugs, the gender pharmacokinetics. So since the 1990s, more women have been included in clinical trials, including PK of new drugs, and that's an advance. However, prior to that time, less between gender PK was studied, and one example is Diclectin. There are many others, of course, of drugs that are out there. Many of the diuretics and everything have not been studied in this way, because at the time there were not guidelines or regulations pushing it. But Diclectin is doxylamine succate and pyridoxine hydrochloride. And it had a difficult history, as some of you may know. I think it even broke one of the companies in Doyle. Or certainly gave them difficulties, Merrill Doyle. Eventually in the US it was withdrawn from the market. Nothing to do with science. Very sadly, this was all the courts that forced this drug off the market. And the general statistics was there were no more deformations in babies with or without the drug being present. So Duchenne has provided me access to some new data that's quite interesting. And that had helped because they had improvements in assay methods, particularly of vitamin B6 metabolites. So the major one that I put up, there were 18 non-pregnant women administered a single dose of Diclectin as two 10 milligram delayed release tablets at night at 10 p.m. on days 1 and 2, and then multiple oral doses from days through to 18. And the schedule was one 10 by 10 milligram delayed release tablet at 9 a.m. and 4 p.m., and then 2 in the evening. This would be the time you would give it for morning sickness, of course, to prevent morning sickness. And it was under fasted conditions at least 2 hours after eating. And by the way, this is 40 milligrams per day and is the maximum recommended daily dose by Duchesne in their product monograph. So what you could do then is compare the first dose area under curve exposure with the final AUC 24 in day 18 to provide an accumulation index. So the new study in these 18 women on that regimen found a threefold accumulation of dexylamine after multiple doses. This is new data. You can see it sort of demonstrated in the graph here also. And maybe that's of concern. But when you look at the metabolites of pyridoxine is really a pro-vitamin because its main active metabolite is pyridoxal 5-phosphate, PLP, and there are other ones, and I have highlighted the PLP here. So the accumulation index is not significant, particularly for the parent, but look at PLP, it's 4 times the high, and some of the other metabolites are 6 to 7 times. So it does demonstrate that the pyridoxine metabolites have clear accumulation after that total daily dose, and some metabolites displayed the 7-fold accumulation, along with the increases in half-life. It's difficult to interpret this because of very complex metabolism of pyridoxine. However, the impact on the safety of patients could be serious, and that's considering that we hear, I believe this is the case anecdotally, that some off-label use of Diclectin is up to 60 milligrams per day. That is not approved by the company's monograph, but so, you know, if doctors are doing that, they're on their own if there are legal suits, really. I've not got time to show other studies, and actually I couldn't get all the data that would have demonstrated this clearly. Certainly, with Diclectin, women tended to be more variable. Intra-subject CV was looked at than men, and there appeared to be a gender difference in the effect of food. That it seemed to be really a formulation gender interaction in that particular effect. But I couldn't really come and nail it down, so I'll leave it as a question. Nonetheless, the accumulation information from the first multiple dose study of this drug in women suggests such information is of concern, especially if the higher doses are being used off-label. So my conclusions, and I'm going to make a very important point at one of these things that should be addressed internationally. Women are being included in pharmacokinetic studies for NDAs in accordance with all the guidances that are out there now. Older drugs clearly are less studied, and few studies in pregnant women are really available except for antivirals and antimalarials. And except for drugs used entirely in one gender, BE studies are supposed to include representative numbers of men and women. But the problem with this now is many of the studies are being done in Asia. And I see in my work quite a lot of bioequivalence studies, they all have the inclusion criteria to have pregnant women, but it is rare from an Asian country to see any women in these bioequivalence studies. And I think this is something that needs to be considered by the agencies. If we are not getting that type of data, it's worse than the situation when we started, really, 10 years ago. So that's a big point that should be thought about. And you look at bioequivalence studies by the FDA, who have the database, to compare the results in men and women might help determine if formulation by gender interactions are a problem. Body weight corrections certainly don't remove all the clearance differences. The questions remain, and I haven't addressed those at all, about the effects of pregnancy, menarche, and menopause, whether certainly that should be studied in pharmacokinetics, perhaps not bioequivalence, but there could be differences in absorption. I think there is certainly less concern about bioequivalence than PK related to effect. That's my own opinion, but I don't say it shouldn't be looked at. And I do agree, you know, if a drug is used only in women, as we find with men, that the bioequivalence should be done in women. I agree with that. Thank you.
Are there any questions or comments for Dr. McGilveray? One question.
Is there a cultural issue why, let's say, my understanding is the first time I hear that is like in India, I guess?
Well, I don't want to name countries, but it's generally offshore, and it's certainly Asia, yeah. So yes,
over
I
there
think
they don't use women?
I think it's cultural. Yeah, women are not. Well, you know that in women, the hospitals are even separate for women, I believe, and so I think that there is a cultural problem, and women don't tend to volunteer over there either. So when they say they'll have a representative population will be studied, that isn't happening anymore, as far as I can see.